Now I'd like to introduce Dr. Brian Wilson. He's professor at the Department of Medical Biophysics at the University of Toronto. Thank you, Professor.
Thanks. Okay, good morning. I will not show you any nasty infectious diseases, so you can relax. What I want to do is to go back to some of the basic science. How do I get this to appear here? Ah, thank you. Of PDT and talk about the issue of singlet oxygen. We have heard about singlet oxygen at various points during this meeting, but I want this time to really concentrate on singlet oxygen and to understand it in more detail. So I will talk a little about the characteristics of singlet oxygen, its role in PDT, and then the actual determination of singlet oxygen, how it is measured, both indirectly and directly. And then I will talk in particular about this direct technique, which we have done a lot of work on, and give examples of the applications of this particular technique. I'll talk about some of the fundamental issues in dosimetry of singlet oxygen, and then some future trends and developments. So what is singlet oxygen? As I think you know by now, this is an electronically excited state of oxygen. The normal ground state of oxygen is a very unusual molecule in that it is a triplet state. Most ground state molecules are in a singlet state, but oxygen is unusual because it is the ground state is a triplet, and in fact, if it was not for that, then PDT would not work. Singlet oxygen, I'm sorry, this should be 1O2. Singlet oxygen is a situation where the unpaired electrons in the molecule are actually in the singlet state. In other words, they are aligned against each other rather than together. Singlet oxygen can be produced not only in PDT, but for example, it can be produced chemically by this sort of reaction. Singlet oxygen is also produced during photosynthesis, so that in a normal photosynthetic process, it is one of the pathways that can occur is actually the generation of singlet oxygen in the photosynthetic pathway. Now, as we know from PDT, because we use singlet oxygen to kill cells, it is not good for plants to generate singlet oxygen. This is essentially a byproduct of photosynthesis. And so plants have evolved many antioxidants in order to scavenge the singlet oxygen that is generated so that the plants do not become destroyed by the singlet oxygen that they themselves produce. And so, as we know, many plants contain antioxidants, and the plants have these antioxidants to protect themselves. But when we eat these plants, these particular vegetables, then we get the antioxidant protection also. So these plants are very kind because they have developed methods to reduce the effects of oxygen species that can otherwise do damage also to us. You know by now that in terms of the photophysics of PDT, there are two main pathways. One is type 1, where the ground state of the photosensitizer absorbs the light energy, is excited to a singlet state, and then that singlet state directly interacts with some biomolecule, some target, and produces some products. Type 2, which is the involvement of singlet oxygen, the ground state of the molecule, the sensitizer absorbs the light, is excited to a singlet state. But now the singlet state undergoes a transformation, intersystem crossing, by spin flipping one of the unpaired electrons into a triplet state. And because oxygen, ground state oxygen, is also a triplet state, the triplet of the sensitizer can transfer energy to the ground state oxygen. That is allowed by quantum mechanics. And so this interaction can take place whereby the energy is transferred from the triplet state of the sensitizer to the ground state of the oxygen. The sensitizer returns to the ground state, and singlet oxygen is produced. Singlet oxygen then goes on to interact with the biomolecules to produce the products. And we'll see this in graphical form shortly. I wanted to also just make the point that although we think of singlet oxygen as the main mechanism in PDT, there can be processes in PDT that are not mediated by singlet oxygen. I already mentioned type 1 processes. However, it's also possible in PDT to produce higher excited states of the molecule. And also there are some unknown processes which, and I'll show you one piece of data, which we don't really understand. The photophysical mechanism that's underlying the process. And that example is an experiment that was actually done here in San Carlos. And actually this is now published. Sorry, this slide is out of date. And the experiment was to take photogem, which is essentially photofrin, and to look at the rate of bleaching. We talked about photobleaching. As you give the light to the photosensitizer, it becomes bleached. And so what was done was that the bleaching rate was examined as a function of the oxygen concentration. Now, if the bleaching is caused by the singlet oxygen, so the sensitizer is activated, generates singlet oxygen, and then the singlet oxygen, instead of attacking cells, there are no cells here, this is just in solution, the singlet oxygen attacks the molecule itself, then you'd expect that the rate of bleaching would decrease with the lower oxygen. Because you have less oxygen, you produce less singlet oxygen, therefore the rate at which the sensitizer is bleached should go down. And that indeed happens. But there is something very strange happens, because at very low oxygen concentration, suddenly you're able to bleach the drug more rapidly. And so that is a very strange phenomenon. It's very well confirmed. This is a very difficult experiment. If you try to do these experiments, and Vanderlee and Christine and I had many arguments about this experiment, this is a very difficult experiment because it's very hard to get solutions down to these very low oxygen concentrations. So oxygen diffuses through plastic and so on, and it's hard to do this, but we're pretty sure that this is correct. And so there's some extra mechanism is coming into play here. And so this is just to illustrate to you that although we'll talk about singlet oxygen as the main process, there are other more complex processes that can also happen. And the question is, what is this and what's the photobiological effect? So just to go back to diagrammatic form, here is a deliberately very complicated plot which shows the molecular states of the photosensitizer, the ground state, excited to the singlet states. There are also, of course, excited singlet states. It's not just one level. There's internal conversion. You can get the sensitizer dropping back to the ground state and emitting fluorescence. You can get the intersystem crossing to the triplet state, and then it's this triplet state that exchanges energy with ground state oxygen to produce a singlet oxygen, produce the biochemical effects, and then the biological and the clinical effects. So once you produce singlet oxygen, what can happen? Well, singlet oxygen is extremely reactive. I'll show you in a moment that the particular reactions that take place are very dependent on the localization of the singlet oxygen. But in principle, there are many, many photochemical effects that can happen. And these can happen in proteins, they can happen in lipids, they can happen in DNA, if you can get the photosensitizer into the nucleus. And in fact, it has been shown that if you can get a photosensitizer into the nucleus, it's about 1,000 times more effective in cell killing than if it does not go into the nucleus. So there are many processes that can happen at the photochemical level, and there's lots of literature on this. I won't go into details. But a very important question is, how long does singlet oxygen last? So once you generate it, for how long in cells and tissues does the singlet oxygen last? This is actually a little controversial. There are actually two different schools of thought on this. The situation is a little unresolved. When you look at the experiments that are done, it's hard to find any errors in the experiments. and yet there are big differences in the results that are obtained. In particular, Peter Ogilvy in Denmark believes that the singlet oxygen lifetime in cells is of the order of several microseconds, while many other people, this was work that we did, this was work that we did, this was some work that was also done in Canada, indicate that the singlet oxygen lifetime is more in the region of a fraction of a microsecond than several microseconds. You might think that that doesn't matter very much. Half a microsecond, 2 microseconds, who cares? But in fact, this makes a huge difference because the lifetime essentially determines the diffusion distance. So once you have generated the singlet oxygen, the question is, how long does it last before it starts to diffuse to other sites in the cell than the site where it was generated? Now, if singlet oxygen lasted a long time, let's say it lasted several seconds, then it doesn't really matter where the photosensitizer is located in the cells. And so we don't need to worry about making a photosensitizer that's targeted to the mitochondria or targeted to the nucleus, whatever. The dangerous material, the singlet oxygen, will diffuse, and it will kill the cell no matter where it was generated. However, if the lifetime is, as we believe, only a fraction of a microsecond, then the singlet oxygen only lives long enough to diffuse perhaps tens or hundreds of nanometers. That means, if this is true, that the location of the photosensitizer is the location of the damage. So if the sensitizer is located in the mitochondria, then it's the mitochondria that are damaged. If it's located in the nucleus, it's the DNA that's damaged. So this is an important question, and there's lots of evidence, and I'll show you some in a moment, to indicate that the diffusion distance is very short because the location of the sensitizer has a big effect on the cell survival. So the subsoil localization determines the site of the primary damage. And the evidence for this is here. These are cells, mammalian cells, in which the cells were exposed either to ALA, and ALA produces PP9. We have heard that many times here, and we've seen clinical examples, and is produced in the mitochondria. In these cells, rather than incubating the cells in ALA, they were incubated in PP9 itself. And if you take relatively short incubation time, then the PP9 is located primarily in the cell membrane. So here we have the PP9 in the mitochondria, the PP9 in the cell membrane. And this axis here is the singlet oxygen, and I'll tell you later how this is measured, but except for the moment that this is a true singlet oxygen that's generated in the cells. It's not the exposure time or the PDT dose. This is a true singlet oxygen, and this is the survival. And you notice that the cell survival in the case where PP9 was given is much less effective, or the treatment is much less effective than if the PP9 is located in the mitochondria. And so this is both tells you that the site of localization is very important, but it also tells you that the lifetime of the singlet oxygen must be short. Because if the lifetime was long, it would not matter where the photosensitizer was located. But nevertheless, you can still ask the question, is singlet oxygen primarily responsible for the PDT-induced cell kill? So I just wanted to run through some pieces of evidence on this, both direct and indirect. So here is the oxygen dependence of cell kill. So we know that for many photosensitizers, this is cell survival versus light dose. And this is a situation where you have very low oxygen. I can't quite read it there, 1.8% oxygen in the solution, and here's 10% oxygen. And this is just the corresponding survival versus oxygen concentration. So clearly, at least under these conditions, the cell kill is strongly dependent on oxygen. Now that does not tell you that it depends on singlet oxygen. There could still be some other reactive oxygen species generated. However, if you add to the solution other molecules that specifically will scavenge singlet oxygen, and so you can, some of the scavenging molecules, such as those in plants, will scavenge any reactive oxygen species. Some of them will scavenge only singlet oxygen. And so, for example, these are curves showing that when you add the scavengers, you get a reduced cell kill. So that if you add a material which, if you like, eats up the singlet oxygen, then the cell kill is reduced. And so indirect evidence that it's singlet oxygen. Now, why is this important in clinical PDT? So we've had several talks on PDT, and this is particularly for cancer, but the same would be true of pathogens. So here are 4 different patients. This was a gentleman who we treated many, many years ago, or at least we tried to treat many years ago, who had nasopharyngeal cancer. He was given photofrin and a lot of light. Nothing happened. 0. We treated him again. 0. So this was a case of complete non-response. He had lots of drug, lots of light, nothing happened. At the other extreme, here is a patient treated by Alan Osseroff in Roswell Park several years ago. This is a patient before treatment. And I'll let you, would anyone like to guess? This is tumor along this strip. Would anyone like to guess what this condition is caused by? Why would you get tumors along a strip? No guesses? So this patient, as a child, received spinal irradiation. This is the exit beam of the radiotherapy. So he was treated by the spine as a child with a long strip of radiation to treat the spinal disease. The beam exited through his abdomen, and 20 or 30 years later, there is a strip of cancer. So this is radiation-induced cancer. This would be extremely difficult to treat. I mean, you can't imagine how this would be treated. You can't do it surgically. You can't take the whole of the front of the person off. This was treated at Roswell Park in a number of patches, and you see 4 years later, this is completely successful. So this is a case where nothing happens. This is a case of complete clinical response. Here's an example from Merrill Beal. These are MRI scans before and after treatment of a large tumor. You see the some opening of the airway here. This was a partial response. This is 2 patients from our own experience with prostate cancer, where we're trying to treat the whole of the gland. This was a very successful treatment. In this case, in this patient, the whole of the gland was properly treated. In this case, it was very inhomogeneous. Same drug, same light, 2 different patients, 2 different results. So clearly, using just the amount of drug and the amount of light as the dose for PDT is not adequate. And in fact, as several people during this meeting have indicated, that lack of ability to properly understand how much dose we're really giving in the patient is probably one of the main limiting factors in the adoption of PDT into clinical practice. So we really, in this case, have an uncertain effective PDT dose. Of course, there are many, many biological reasons why there may be these differences. For instance, in this gentleman, we undoubtedly gave a huge drug and light dose. And so either there was absolutely no oxygen in this tumor, or there was some biological reason why this tumor was completely resistant. But certainly there is some of these variability is due to the uncertainties in the amount of PDT dose actually given. And those groups who have worked on PDT dosimetry for many years, you might ask, well, why is the problem not solved? Why can't you just give the clinician the instrumentation to do the dosimetry and let them get on with it? Well, it turns out the reason is partly that we're not very competent. That's one possible explanation. The other explanation is that this is a very, very complicated problem. Because of the fact that there's light, sensitizer, and oxygen, these are three different parameters that come together to produce a singlet oxygen. But you have to realize that these are not independent. These three parameters of light, drug, and oxygen are not independent. So that, for example, the light plus the oxygen bleaches the drug. The light plus the drug deplete the oxygen. If there's a lot of photosensitizer, you attenuate the light. So these are all interdependent parameters. Not only are they interdependent, they change dynamically during the treatment. So for example, if you give the treatment and you reduce the amount of oxygen, either because you photochemically use it or you have vascular shutdown, then you will reduce the rate of photobleaching of the photosensitizer. So this is a dynamic, complex process, and the question is, how do you get a handle on this in order to finally get a description of the real dose here that leads to the biological response? And we know that the biological responses themselves can be very complicated. It can be direct cell kill, effects on the vasculature, effects on the immune system. So the biological effects are complicated. The dosimetry is very complicated. And this is one of the big challenges in PDT. So what we would really like to do is, if it really is true that singlet oxygen is responsible for the biological effect, then in principle, whatever we choose as a measure of response, whether cell kill or vascular effect, at least the direct response, you might think that if we plot the response versus the singlet oxygen, we should be able to generate a curve. And that curve is essentially the biological sensitivity. So we have a very steep curve, then we have a very good photosensitizer. It's located at a very sensitive site. The tissue is sensitive to the treatment. If we have a very flat curve, then this is a very low sensitivity situation, and maybe PDT is not the best thing to use. And the big question is, how do you measure that? And there are three essential possibilities. You may be able to think of others, and that would be great. The three are direct and indirect, and there are two different types of indirect. So I'm going to walk through each of these in a little more detail. So the direct technique is by near-infrared luminescence. If you go back to the energy diagram, we have the ground state sensitizer, absorbs the photon, excites to the singlet, crosses to the triplet exchanges energy with oxygen, generates singlet oxygen. Now that singlet oxygen can itself go back to the ground state. It's an excited state. So just as this sensitizer can go back here and give off fluorescence, the singlet oxygen can go back to molecular oxygen and give off luminescence. That luminescence happens at 1270 nanometers. This is a well-known phenomenon. There's no question that this happens. So one possibility is you measure this light. So we measure the 1270 nanometer light, and that will give you the direct measurement of the singlet oxygen. And this shows, I'll show you in more detail how this is done, but this is a setup in our lab. Here's a volunteer mouse from our lab, and we irradiate that with a laser beam, not the treatment beam, but a separate laser beam to measure this singlet oxygen. I'll tell you how this works. The challenge in this is that this decay back to the ground state, and emission of singlet oxygen happens only in 1 in 10 to the 8 singlet oxygen molecules. So only 1 in 100 million molecules of singlet oxygen goes back to the ground state. The reason for this is that singlet oxygen is so reactive with biological materials that all of the reactions, except very rare, happen to the biological interaction. If you take a cuvette of sensitizer in water, then this signal is much, much higher. But in tissues, it's a very low signal. The second possibilities are indirect. So one is that rather than directly try to measure the singlet oxygen, can we use another substance, another molecule, which reacts with the singlet oxygen to produce a product that we can measure? And in particular, an optical signal such as fluorescence or chemiluminescence or a color change to this molecule. And I'll show you an example of that. And the third possibility is that if the sensitizer photobleaches, so if the singlet oxygen interacts with the sensitizer and bleaches the sensitizer, if we monitor the sensitizer by, say, fluorescence, then we measure a reduced fluorescence, and perhaps we can calculate from that reduction of fluorescence how much singlet oxygen was generated. And this just shows an example here. Here's a patient. This is in the lung. Here is the sensitizer fluorescence image before treatment. And after treatment, you see all of the photosensitizer has been bleached. And if we knew something about the photophysics of the sensitizer, maybe we could calculate how much singlet oxygen was actually generated. So let me start with the middle one of the secondary reporter. So here's a couple of examples. This is indirect imaging of singlet oxygen using this molecule, SOSG. And this molecule is not fluorescent until it is attacked by singlet oxygen, and only singlet oxygen, and it becomes fluorescent. And this you see here. Here is cells incubated with PP9, and as the PDT exposure goes along, you get more and more of this fluorescence, and so this essentially reports the singlet oxygen that is generated. And in a situation where you do not generate singlet oxygen, the conditions are set up so this doesn't generate singlet oxygen, you do not see any signal. So this is very nice. You think that's a good thing to use. What's the limitation of this? So we want to do this in the clinic. We're going to give this material. Let's assume it's not toxic. Maybe it's toxic. We'll give this material to the patient along with the photosensitizer, and then we'll just look at fluorescence, and we measure the fluorescence, and we'll use that to measure the dose. One disadvantage. Yeah. So another background source of fluorescence, yeah, that's one possibility. You might address that by, for example, you could look at the spectrum of the fluorescence, and maybe that will give you, yeah.
Okay. So most oxygen interacts with biological tissues and biomolecules, not with our sensor.
That's true, but you see from here, it looks as if, at least these are reasonable conditions, it looks as if there's enough reaction to give you a signal. Maybe that in a patient you would get a very low fluorescence signal, but that's another possible limitation.
It might not get into the tumor or the same location.
Right, and that's the third possibility, is that here it's easy because the culture is incubated with the sensitizer as well as the reporter, so we know that it's locating, whereas in a patient perhaps this molecule would not co-locate with the photosensitizer. So it's a very nice idea. It has not been used in patients to this stage. It has been used, for example, to study singlet oxygen damage in plants. So these are some very, I put this up just because they're very pretty pictures. So these are images of secondary fluorescence reporter in plants as the singlet oxygen is generated under different conditions. This is a very powerful technique, actually, for looking at singlet oxygen in plants. Another possibility is to use chemiluminescence rather than fluorescence. So here we have the singlet oxygen, a reporter molecule, and now the reporter molecule becomes chemiluminescent and gives off light. And this has been shown to correlate very nicely. You measure the chemiluminescence light, and that correlates very nicely with cell counts. And actually, this is strong enough to signal, to image. So you can actually make it. This is an image of the chemiluminescence that's generated by singlet oxygen. So you know in this case, in this particular tumor, you are generating singlet oxygen because of the chemiluminescence reporter. Again, the same problem of co-localization, perhaps not enough concentration, perhaps although the background problem here is not a problem because there's very, very low chemiluminescence background. There is a bit of a problem because this is probably toxic. So that's a limitation, at least in clinic, although it's a very nice tool for preclinical studies. So the reporter molecule, you have nice strong signals, it's relatively cheap to do, it's fast, you can do imaging, but it does depend on these other factors. And actually it turns out quantification is difficult because really, if you want to know, if I go back to this, you can certainly measure how much chemiluminescence is produced, but if you want to say, okay, how much singlet oxygen does that correspond to, you have to know the concentration of the chemiluminescence. You have to know this concentration in the target. So it moves the problem to a different problem. But for preclinical studies, these are very nice techniques. So how about this one? We're going to do this. We're going to apply some mathematics and calculate the singlet oxygen from this photobleaching. This has been done very elegantly by Michael Patterson's group in Canada, where they set up the equations that describe this process of the singlet oxygen generation, and then the singlet oxygen interacts with the photosensitizer. And so you can calculate from this an equivalent dose, and in fact you can essentially calculate the singlet oxygen generated. And if you do this in cells, this is a singlet oxygen dose calculated looking at cell kill, and it works very beautifully. I don't have time to show you, but this approach works well until you start to consume significant amounts of oxygen. And then things start to fall apart a little. But in principle, this is a very nice technique, and of course could be used clinically because you could just measure the fluorescence. So Dr. Allison and Dr. Hopper emphasized in the clinic the value of measuring fluorescence, but you should not only measure the fluorescence to tell you how much sensitizer is there, you should measure the bleaching of the fluorescence to tell you if you're delivering enough dose. So this is nice. The photosensitizer is itself the reporter, so it does not need another molecule. You have a nice strong signal, relatively inexpensive, imaging possible and fast. It is dependent on a model, so you need this photophysical model, and models are always as good as the assumptions. It does depend on the sensitizer, whether or not the model is correct, and it may break down under low oxygenation. So for example, this is a plot of fluorescence. This is done actually in skin versus the light dose, and you see that the photobleaching rate in this patient actually suddenly changes. And so that tells you that if we applied this model, it does not describe this behavior. So it's simple to do in practice. It's a little more challenging to be certain that the numbers you calculate are correct. But if you know enough about the photophysics, then this is a good technique. So let me come to the third technique of the direct monitoring. So again, we have the choices of in photosensitizer, the symmetry of measuring light, sensitizer, and the oxygen, or we measure the singlet oxygen directly by this emission. It avoids the complexity of trying to measure these other factors independently. And I think Dr. Zhu will probably talk about some of the, Dr. Zhu's talked about dissymmetry for light yesterday or the day before. And there are techniques for measuring fluorescence and for measuring oxygen. This avoids the need to measure these things independently. If you look at the kinetics of this, what is happening is that the triplet state process is generating the singlet oxygen, and then the singlet oxygen is decaying, both by this process and by the biological effects. So this is equivalent to filling up a bathtub where the plug is removed. So you turn on the taps, and you generate lots of water. You generate lots of singlet oxygen. So this is turning on the tap. But the water, the singlet oxygen, is draining away from the tub. So the question is, how much water is there in the bath? You know, and so it depends on the balance between these two processes, and that is just described by this equation. One of these terms fills up the bath, and the other term empties the bath. And so it's the balance between these two that determines how much water, how much singlet oxygen is actually generated and produces the PDT effect. And you can write this down as an equation, and you can look at the curves as a function of time, as the following a pulse of light. What is this signal? Now, the important point here is that the signal is proportional to the concentration times the singlet oxygen lifetime. And I just told you earlier that the singlet oxygen lifetime is very short, and in practice, only about 1 in 10 or 100 million emits a luminescence photon. So this signal is very weak. The second problem is a technical problem, is that 1270 nanometer is a horrible wavelength to detect light. And this is the fault of the physicists and the engineers. They're not clever enough to invent good detectors. Actually, it's not really their fault. There's some fundamental laws of nature that make this difficult. But essentially, if you look at conventional detectors, they run out of steam at this wavelength. And also, of course, we are hot, and we generate black body radiation. This is just a thermal image. This is actually used clinically sometimes. We are giving off near-infrared radiation all the time. And so you have a horrible biological background that you have to deal with. So a little history here. The first report of using direct singlet oxygen luminescence was in 1987, and this shows the peak at 1270 nanometers. Very, very noisy signal. It was done in tumor. However, this could never be replicated. No one was ever able to reproduce this result, so it's still in the literature as unknown how John Parker managed to get this result. There's some theory around why he got it, but there's it's not clear. And so until 2001, it had never really been confirmed that you could measure the singlet oxygen in cells or in tissues this way. There were some early studies, including ones that we did 23 years ago, where we used an existing detector for the physicists and engineers here. This was a germanium detector, and we got no signal. So it was a completely negative study. We still published it, because although it was a negative study, the fact that we did not get a signal allowed us to calculate the upper limit on the lifetime. Okay, so if the singlet oxygen had lived long enough, this would work. The fact that we got no signal tells us the lifetime must be less than, in that case, 500 nanoseconds. A number of groups, particularly in Europe and in Russia, did measure singlet oxygen in cells, but they did it by a trick, because it turns out that if you grow cells in D2O instead of H2O, you significantly increase the lifetime. And so, since I showed you the signal is proportional to the concentration times the lifetime, they were able to get a signal. But of course, you cannot expect to convert a patient from an H2O life form into a D2O life form. So this is very nice research tool, but again, it won't move to the clinic. The breakthrough was a technological breakthrough when Hamamatsu brought out this photomultiplier tube, which this is a normal photomultiplier tube response. This is wavelength versus quantum efficiency, and they managed to produce a photomultiplier tube with extended near-infrared. This was not generated for singlet oxygen dosimetry. No one can make money out of doing singlet oxygen dosimetry. This was generated for long-distance telecommunications, for repeater stations in long-distance telecom. But nevertheless, this was a nice tube. It has quantum efficiency of about 1%, and please remember that number. This was considered at the time to be extremely good, but I'll come back to that. So you set up this experiment. You want to measure the time curve, and so you have a pulse laser, about nanosecond pulses, operating at about 10 kilohertz, and you essentially come down into the sample, and then you have to remove a lot of the background, fluorescence, etc. So you have a filter wheel, and then the photomultiplier tube, and then you have the specimen, and you can measure the spectrum, and you can measure the time dependence. Here was the very first proof of this technique. This was in cells, just looking at the wavelength dependence with a couple of different concentrations, and this was the first in vivo measurement in skin. Here's the control animal with no photosensitizer. Here is with the sensitizer. You see this is not a big signal. And if you look at the time curves, in fact, the differences are pretty subtle in the time curves. One of the problems is you can't really use the early part of the curve because it's dominated by fluorescence and other sources of signal that you cannot get rid of. So we have that technology prove that it actually is working and measuring the singlet oxygen. You shouldn't believe that just because you built it, that it actually works. You can get signals, but is it a true signal? So is the signal really coming from singlet oxygen? So here's some experiments that were done to try and prove this. This is all done in photosensitizer in solution. So here is what happens if you add protein. So if you take a photosensitizer in a cuvette and you measure the singlet oxygens generated, what would you expect to happen if you added protein? And so this shows you the change of the triplet state and the change of the singlet oxygen level. So that was consistent with theory. What happens if you take cells and you incubate them with the photosensitizer and you wash them so there's no photosensitizer and you measure the singlet oxygen, and this is what you get. Now, if you allow the cells, this is what you get, the red curve. If you allow the cells to sit there for any length of time, like a minute, in the cuvette, and you make the measurement again, you get a much bigger signal. And this is because the sensitizer, a little bit of the sensitizer leaks out into the water. There's no biological substrate in the water, therefore the lifetime of the singlet oxygen from the photosensitizer that is in the water is longer, so it contributes to the signal. So again, these experiments are easy to do, very difficult to interpret. You have to be very careful. And then finally, what happens if you drop some sodium azide, which is a singlet oxygen quencher, into the solution? You get this death of the signal. And so this is probably the most unarguable result, namely if you take the sensitizer, measure the singlet oxygen, put a drop of sodium azide, which only interacts with singlet oxygen, you immediately kill the signal. So this is it's pretty difficult to think of another reason why this would behave like this. But the important point is that if you're going to do this type of measurement, you need to be very careful about the controls studies that you do. It's easy to get a signal, It's very difficult to get the correct signal. Does it really correlate? Does the biological response really correlate with this signal? So here's an experiment that was done. So these are tumor cells. They happen to be leukemia cells because you can grow them in high density. It doesn't matter. And we used MTHPC. And this is survival curves versus light dose. And these are different experiments. And these experiments, we varied the power, for instance, from 10 to 50 milliwatts per square centimeter. We varied the amount of photosensitizer. We let the oxygen be anything it liked. So we didn't care. So this is an uncontrolled experiment. This is just like what happens in a patient. Doing a PDT treatment is an uncontrolled experiment in a patient. You have no idea what's happening in the tumor. So this looks a mess. So if you were treating a patient, you said, well, I think 25 joules per square centimeter is the right treatment. Well, in this patient, maybe it would work. In this patient, maybe this is a man with a tumor in the nose. Right? Nothing works. If you measure the singlet oxygen for each of these conditions, so this is a light dose versus the total singlet oxygen is generated, you see these curves are also all over the place. So the different conditions generate different amounts of singlet oxygen. So that's survival versus light, singlet oxygen versus light. The obvious thing to do is to take these data and replot survival versus singlet oxygen. And if you do that, you get this beautiful curve. So all of these data that are completely chaotic collapse onto a single curve. And in fact, from that signal curve, you can fit this to an exponent, and you can actually calculate how many singlet oxygen molecules it takes to kill a cell. And so the 1 over E, cell death, requires about 10 to the 7 molecules of singlet oxygen per cell. Does that sound like a lot or not very much? Right? It's actually quite a lot of singlet oxygen to generate per cell. And this, of course, is for photosensitizer MTHPC, so this is probably located in the mitochondria. But it gives you, for the first time, a number that is a true number about the sensitivity of the cells to the treatment. Does this work in vivo? The answer is yes. The model used for this was topical ALA in skin, and we've heard lots of clinical work on topical PDT in skin. You can measure the signal. It's a very small signal. And here we're giving it the ALA, waiting for some time, and then giving a dose of light, and we measure the response, the skin response. So here's the skin response. So the higher the number, the more severe the response, versus days following treatment. And there's a paper in the literature from the group in Rotterdam showing that if you gave the same amount of light but gave it at higher fluence rates, so this is 50 milliwatts per square centimeter, 150 milliwatts per square centimeter, you get much less response for the same amount of drug and the same total amount of light. So again, this is singlet oxygen versus light, and this is the response. So what happens if we plot the response versus singlet oxygen, and we get a nice curve. So this tells you that it really is the singlet oxygen that's producing this biological effect. So if you had a way to measure the singlet oxygen in a different patient, from patient to patient, you could actually adjust the light in order to give the right treatment. So this singlet oxygen luminescence dosimetry correlates with the biological response in cells and in vivo. Does it work in tumors? The answer is yes, although it's more difficult. This is the experiment that was done. We used bioluminescence in this case as the response monitor, just as an endpoint. So this was a bioluminescence in the tumor before treatment and after treatment. This is a window chamber model where the tumor is grown in this chamber. We can actually image or do crude maps of the singlet oxygen, and ignore this curve for the moment. This is response as measured by bioluminescence versus the singlet oxygen. You see there's a lot of scatter, as you would expect for tumors, because no two tumors are the same, even if they're from the same cell line and injected in the same animal, but you get a response curve. So that seems to work. And a group in Japan has also done this a number of years ago, looking at brain tumor in vivo. So if you believe, therefore, that this singlet oxygen dosimetry system really works, then you can apply it as a gold standard. And a number of studies have been done using the singlet oxygen, this SOLD technique, as the gold standard. This is an example of molecular beacons. And the idea here is you take a sensitizer, a quencher, you join them together by a linker, which can be cut, for example, by an enzyme which only exists in the tumor. This was originally done for imaging a number of years ago by Ralph Weisleiter. So this is an image of a molecular beacon. This is really an image of the enzyme activity in the tumor. And you see that this is the singlet oxygen before the beacon is cleaved, and then suddenly, after you cleave the beacon, you free the sensitizer, and you can generate a singlet oxygen. And this works not only for enzyme-cleaved beacons, but also for these so-called hairpin loop beacons, where when you open the loop, because the strand of DNA couples to the complementary strand in the cells, then again you get the singlet oxygen signal. We did an experiment with Barbara Henderson at Roswell Park, looking at whether or not it correlates with a particular molecular signal. And this molecular signal was a STAT3, which is to do with cross-linkage. And again, we showed that you can actually correlate the STAT3, a molecular signal, with the actual singlet oxygen measurement. And it's been also applied to nanoparticle PDT. I showed you this a couple of days ago. You can measure the singlet oxygen generated from here. And this has been done also using quantum dots. The limitation of this technique is the signal is very weak, as I've already told you. There's lots of technology problems to do with this that I don't have time to go into detail. I just wanted to point out that fiber optic based detection is very difficult, and I'll come back to that. At a more fundamental level, if you think about what is happening here, we are measuring the signal that comes from the decay of the molecule. But that means that the signal is coming from the singlet oxygen that does not interact with the tissues. So that's a very peculiar measurement. Right? We want to measure the interaction of our agent with the target, and we do that by measuring the agent that doesn't interact with the target. That seems odd, right? So you have to think that's a funny experiment to do. You notice it's integrated over both sensitive and non-sensitive cell and tissue components. So if the sensitizer is sitting not only, say, in the mitochondria, but in the cytoplasm, we cannot differentiate the singlet oxygen that comes from mitochondria from that comes from cytoplasm. The singlet oxygen in the cytoplasm probably does nothing. And then there may be non-reactive species, especially at low concentrations of oxygen, as I showed you. I'm going to skip this just in the interest of time, but I want to point out that there are some data in the literature, and this is a study from Tom Foster, who is an extremely rigorous scientist, so you can really believe these data. And Tom did some studies and I'll just give you, the bottom line is this last statement. Any PDT dose metric that's proportional to singlet oxygen will fail to predict the tumor response. So Tom says, on the basis of what he studied, that this is a complete waste of time. Yeah? And it doesn't matter whether you do it by direct measurement of singlet oxygen or by measuring the light and the drug and the oxygen and calculating the singlet oxygen. This is a complete waste of time. And the argument, and there's some real data, is that it's the micro distribution of the damage that matters. So making a volume measurement of the response and a volume measurement of the cytotoxic species will not work. If that's true, it's a bit despairing. It means we're really stuck without any means of dosimetry. But I want to just warn you not to be over-enthusiastic about the possibility of dosimetry. Just to come to a close, I want to point out that some of the technological issues here, for instance, in the system that we and others have used, you need these nanosecond pulse lasers. that are at sort of kilohertz frequencies. You can't have them too rapidly pulsing, otherwise the signal from one pulse bunches up against the signal from the next pulse. And it turns out that these lasers are actually not very accessible because it's not really possible. We have not been able to find any such laser except if you spend $200,000 that will work, that will give you red light. So a challenge to the optical engineers, please develop a red nanosecond kilohertz laser that we can afford. This group, PSI in Massachusetts, said, well, let's accept that. Let's use a diode laser. And let's just pulse the diode laser. Now you say, it's not fast enough. You know, the fastest I can do this is maybe a millisecond pulse length. So I said, okay, live with that. But the edge, the trailing edge of this is still very short. So why don't we just ignore all this signal and just measure the signal coming from this trailing edge? And that's indeed is what they've done. Here is the signal. Here's the pulse. And we're just interested in this trailing edge. And so if that worked, it would be very nice. There is a study in the literature from Mass General Hospital where they've measured this actually in humans with ALA, similar to the mouse experiment. And they've measured the score, the edema, versus the Singlet Oxygen change. It looks like that's very nice, but I would point out, if you look at that paper in great detail, it's not clear that this is only measured in Singlet Oxygen. If you go back to all the earlier studies I reported, it's very difficult to be sure that you're measuring just Singlet Oxygen. I personally don't think this is working, but that's a bias, and I've argued this with the people from PSI and Tayaba, so we have an argument, and that's fine. I don't think this works, but maybe it does. Hopefully they're right and we're wrong. Coming back to this, I said this was a big breakthrough. We had this quantum efficiency of 1%, but it's only 1%. And we're chasing signal. Is there a better detector? And so we very recently did an experiment in Edinburgh, in Scotland, with Robert Hadfield. And a number of groups, not Robert's group himself, but a number of groups, including a group in Russia and another group in Europe, have developed these new detectors, which are superconducting nanowire single photon counting. This sounds very exotic. Right? And actually it is pretty exotic. This they take, you know the phenomenon of superconductivity, that if you take particular metals down to a very low temperature, then they have zero resistance. And so they take a nanowire, tiny little wire of superconducting material, take it down to this, the critical temperature. In fact, it's very important to hold it just at the critical temperature. And now what happens is that a photon comes in and is absorbed by that nanowire. That photon provides energy. And it gives an instantaneous little increase to the temperature, which makes the nanowire go non-critical. So it becomes above the critical temperature and becomes resistant and produces a voltage pulse. In this case, the quantum efficiency of this is very good. This is the essentially the quantum efficiency. That's up to about 15% at the moment, and Robert has told me that he thinks that they can get this up to 100%, or close to 100%. This is just to show you that I sometimes do go into the lab, very rarely, but this is the detector. This is not a small detector, right? It's a nanowire, but all this stuff is the cryogenic cooling, because you have to hold this just below the critical temperature. In fact, if you think about it, one photon comes in. So this is like 2 electron volts. That's enough energy to make this detector go non-critical. So it has to be pretty precise. Anyway, it does work. We set up this system. we measured it. It works. If anyone is interested, I'm happy to talk about it. We did the standard things. Here's the decay curves. Here's the spectrum. Here's the sodium azide quenching. So we're sure that we were measuring the singlet oxygen. Why is this important? Well, the high photosensitivity, or the high quantum yield here, plus the design of the detector, means that we think, in fact, we report in a paper that's just come out, we think that you could actually use a fiber optic to detect the singlet oxygen. So in principle, we think you could put a fiber optic into the tumor and measure the singlet oxygen. And that would be very nice to do. So we're trying to find some funding to do that. What about imaging? I showed you that can be done with D2O. We've done imaging, sort of imaging, just by scanning the laser beam and producing these rough maps, pretty crude. There are possibilities, for example, this technology, the focal plane arrays that have good quantum efficiency out here. The problem is they're extremely expensive. I think Princeton quoted me like $170,000 for this. Our colleagues so it's enormously expensive. This is a byproduct of the military research. These are not developed for singlet oxygen. They're developed for targeting. Our colleagues at Fujian Normal University in China have recently used this cooled camera, which is somewhat more affordable, and have shown that you can actually do singlet oxygen imaging. So this is pretty nice. At least in an animal model, it starts to move into reality. So coming to the summary, the singlet oxygen is direct, is independent of model, and you can make it absolute and quantitative. The signal is very weak, expensive equipment. It has been difficult through fiber optics, is slow, and imaging is challenging. So the conclusions are singlet oxygen measurement is a useful tool for quantitative assessment of pediatric response. Within its known limitations, it is a robust direct PDT dose metric. It's known to correlate with tumor response. Be careful, correlating is not the same as predicting. We have not yet, and no one has done an experiment where they vary the parameters, measure the singlet oxygen, and predict the response. And that's what you want in the clinic. So this experiment still has to be done, even if there's high biological heterogeneity, and this is Tom Foster's point. If there's a lot of heterogeneity, maybe this will not work at all. Photobleaching can be valid and is clinically practical, but you need a full understanding of its limitations, and you have to understand it for the particular photosensitizer. There is not a general metric here that would tell you for any photosensitizer. So if you want to use photobleaching as a dose metric in PDT, you have to do the experiments first with the particular photosensitizer. We've also, sorry, this has nothing to do with this talk. This talk just talks about OCT as a means of monitoring the response. So thank you very much. I want to acknowledge the people that we worked with over the years on this and the sources of funding. I come away from this still enthusiastic about singlet oxygen luminescence detection. I think it's not impossible that we could get this into the clinic. If we could get it into the clinic, I think it would be enormously important to try to understand why we're seeing responses in some tumors, why we're not seeing responses in other patients. And if we do that, even if it never became affordable as a routine tool, we could at least start to understand what is going on in the real clinical world and may be able to devise other techniques which would allow us to develop practical solutions. So thanks very much.
Professor, are those secondary reporters you have mentioned really specific for singlet oxygen production and not for general ROS?
Yeah. Well, that's the claim. The SOSG is supposed to be singlet oxygen specific and does not respond to other ROS. The chemiluminescence, I'm not quite so convinced about. being purely singlet oxygen.
Yeah, that's
but
because
the SOSG is claimed to be purely singlet oxygen. I don't know if anyone's done an experiment to really test that.
yeah, need to cross it with the luminescence signal of 1270,
Right.
maybe, right, to validate that.
Yeah.
Because I was using DCF age and RNO to measure ROS production, and was first mentioned to be specific for singlet oxygen, but it's not
It wasn't.
at all.
Yeah.
Okay. Thank you.
You're welcome.
Thanks, Brian. That was a really great summary. It took me back many years into the into the early days of the hunt for the singlet oxygen.
Mm-hmm.
We've seen lots of changes in conventional chemotherapy, in perfusion and metabolism at tumors. And many years ago, we did some of that work in PDT. And I just am wondering, are any clinical groups really following up on that? Because, of course, with the generation of singlet oxygen, there's the depletion of ground state oxygen. And once that's depleted in the reservoir of the tissue, then oxyhemoglobin needs to give it up. And so there should be the formation of deoxyhemoglobin. You should also see metabolic changes within the cell as a result of the oxygen depletion that perhaps could be detected by looking at mitochondrial cofactors, looking at NADH and FAD, fluorescence, and so forth. So is there any indirect metabolic measurement activity going on to try to really look at kind of the functional changes in the tissue?
I mean, I think it's a nice point, Bruce. The answer is not very much. There was some activity around those areas, I don't know, 10 or 15 years ago. I guess people somewhat gave up because the results didn't seem to be very reliable. However, I do think it would be worth revisiting those. I mean, I had the graph of response versus singlet oxygen, right? I do think it would be worth revisiting some of those more subtle biological response now that there is at least some technology that allows you to measure the insult. I mean, I think a lot of the problems with the earlier studies is, as I showed on that set of chaotic curves, is that you can't control all the parameters. And therefore, if you just use, say, the light dose or the drug dose, whatever, you're going to produce very unpredictable data. So especially if you're measuring a subtle endpoint and you don't really have the x-axis pinned down, then it's hard to interpret. I do think it'd be worth revisiting some of those with the ability to directly measure the singlet oxygen.
Brian, it's a great talk you have on the singlet oxygen. So I have two questions. Both of them are, just one of them is concentrated on your so-called photo bleaching calculation indirect methods. So when one has to do this kind of calculation, there's a lot of photochemical parameter needed, such as lifetime of fluorescence, lifetime of photo bleaching, phosphorescence lifetime, inter-system cross-section lifetime. The one question I have is these parameter currently, what's the state of the knowledge about these parameter for some of those common photosensitizers? And this.
Let me answer that first.
Okay.
I think you're quite right. You have to do. This is why I made the point that if you're going to use photo bleaching as the metric, you have to understand the full process for that photosensitizer. So that if you look at the work that Mike Patterson did with MTHPC, that was 2 years of work to actually pin down all the parameters that were happening there and get all the numbers that you need to plug into the calculation. But it's just work, right? It does not require any conceptual breakthrough. You just have to do the work. Right, so someone does the work with, say, MTHPC, someone does the work with PP9, et cetera. You just have to do the work. And once you've done it, that should be reliable from then onwards.
So the second question is, the SOLD signal you're getting, do you see a variation of lifetime itself in different environments, such as in vivo environment, in tissue, in tumor, or do you expect to be different compared to, for example, in ex vivo cell studies, which, you know, a lot of work has been done to derive some of these parameters?
Yeah, I mean, the lifetime certainly does vary. That has not been studied in any systematic detail, largely because the measurement of the lifetime is not super reliable because of the problem that you cannot use the early part of the curve. And so if you have a curve like that, and you want to measure the lifetime, really you would like to fit the whole curve to that equation with filling the bath and emptying the bath. If you've only got the sort of trailing edge of the curve, that's a little unconstrained. And so it's actually quite difficult to get an accurate measurement of the lifetime. But within those errors, it has been demonstrated that the lifetime does depend obviously on the rate of interaction with the biological substrate. But to my knowledge, Tim, no one has exploited that yet for the interesting question, which would be, let's use that change in lifetime and understand something about the biology.
Thank you.